Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stoliarov II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization. Greetings, ladies and gentlemen, and welcome to our U.S. Transhumanist Party Virtual Enlightenment Salon. Today is Sunday, March 8th, 2026, and we have a fascinating conversation in store for you regarding telomere biology and the potential to reverse biological aging. So joining us today, we have our distinguished panel of U.S. Transhumanist Party officers, advisors, members, including our Director of Visual Art, Artemon Garcia, and our biotechnology advisor, Dr. Bill Andrews, who is also the founder and CEO of Sierra Sciences, regular guest on our salons, and his dog Dash, who is an ultramarathon runner as well, along with Dr. Bill Andrews. And soon to be joining us will be David Wood, who is the executive director of the Longevity Escape Velocity Foundation and the founder of London Futurists. Our special guest today is an individual whom I have wanted to have as a guest on our Virtual Enlightenment Salon for a long time. Dr. Michael Fossel serves as chairman of the board of Telocyte, a biotech company developing telomerase therapy for Alzheimer's disease, with FDA-sponsored human trials planned to target the underlying disease process. A recognized expert in aging and telomere biology, Dr. Fossel has authored over 100 scientific articles, book chapters, and books, and has lectured internationally, including at the National Institutes of Health. His 2020 article, A Unified Model of Dementias and Age-Related Neurodegeneration, generated significant global interest, with more than 1,000 reprint requests presenting both a comprehensive understanding of dementia causation and novel therapeutic approaches. His companion work, A Unified Model of Age-Related Cardiovascular Disease, similarly explores potential interventions in cardiovascular disease. Dr. Fossel earned his Bachelor of Arts and Master of Arts degrees from Wesleyan University before completing his PhD in neurobiology and MD at Stanford University, where he taught neuroanatomy and experimental design. He subsequently focused on aging, particularly premature aging syndromes and the experimental testing of aging theories. For nearly 3 decades, he served as clinical professor of medicine and held leadership roles, including director of the American Aging Association and editor of several medical journals. So, Dr. Fossel, welcome. It's an honor to have you here. And today, the focus of our discussion will be your recent book, which came out in January of 2026, entitled Reversal: Science, Medicine, and a Future Beyond Aging. This book is available on Amazon, where you can order it at this link that I am showing right now. You can also visit Dr. Michael Fossel's website at michaelfossel.com. So, Michael, we're pleased to have you here. Tell us more about the book Reversal, some of the background behind it, what motivated you to write it, and the key topics that it covers.
I am really honored and pleased that you had me. Thanks very much. And it's nice to see my friend Bill here too. The book, in some sense, is something that's been on my mind for 50 years. I wrote a book very similar to this 30 years ago and have written a number of textbooks since, but I'm sort of bringing it up to date and bringing it into what's going on now. I think maybe the key issues start with what I call shortgevity versus longevity. And that is, most people, when they're talking about longevity, they're talking about a few more years, slow the incidence of osteoporosis and osteoarthritis and Alzheimer's. And we're not talking about that. We're talking about actually reversing it. To give an analogy, if I go back 200 years, we didn't know anything about microbial disease, and let alone how to cure it. And we've come a long ways. The current estimates are that less than 1% of people will die of infectious disease in Iceland this year. That was, up in the 80%, particularly for young people, if you go back 200 years ago. Things have changed dramatically. And we're not talking about symptomatic treatment, that is, the book isn't. We're not talking about symptomatic treatment of aging or slowing it slightly. We're literally talking about reversing it. So that's a different way of looking at it. We're talking about dramatically extending human lifespan and preventing and reversing essentially all age-related diseases. That's what we're aiming. The second theme, I think, is an idea that Bill and I have known about now for decades. And I got intrigued by this now 50 years ago because I found that people always had this blasé idea about aging. Aging just happens. It's just entropy. It's wear and tear. Things fall apart. What do you expect? And I've noticed that whenever people are that blasé, it usually means that there's more to it than that, and they're not thinking very carefully. And to put it bluntly, aging is not just entropy. It's a failure of maintenance in the face of entropy. That's a conceptual shift most people don't make. And it takes us down to the kind of thing that Bill and I have been working on now for many decades. But it's a conceptual difference. If I have a friend of mine who's had a car for 50 years. He doesn't have a car for 50 years because it has the best genes, that it was the best car. It's because he maintained it in the face of entropy. The same is true of houses that have been around for 500 or 1,000 years. It's not just that they're well built, but well maintained. And if you think about it, every single cell in your body is not just several decades old, but you got it from your mother, who got it from her mother, who got it from her mother. And that provenance goes back 4 billion years. And the same is true of your mitochondria. It's all well and good to say mitochondria cause aging, but then the immediate question is, what caused mitochondria to age? And the mitochondria have been around for 2 billion years, give or take. So you can't just wave your hands and say, well, it's entropy and it's mitochondria, without explaining why entropy works at different speeds in different species and didn't work retrospectively in the germ cell lineage for 4 billion years. You have to explain that. I don't mean it can't be explained, but you can't just wave your hands and say it just happens. Aging doesn't just happen. And what I noticed, as I said in the book also, is that back 50 years ago, there was a sort of a blind man and the elephant problem. If you looked at the literature back 50 years ago, or even 40 years ago, people were saying aging is this, aging is that, aging is reactive oxygen species, aging is a million things. But there was no single elephant where you could put them all together and say, how do they all happen? And I still see that. I was leading a couple of global conferences on age-related disease. And a lot of people got up and said, all age-related disease is caused by mitochondrial dysfunction. Your mitochondria are dysfunctional, mine are too, over the last few decades. But what caused the mitochondrial dysfunction? And about a third of the speakers got up and said it's because of inflammaging. All right, what caused the inflammaging to occur? It's going back those steps and putting the elephant together that was important. And it wasn't until, Bill and I think Bill was just saying he ran into me about 93. I think it was around 91 or something when I first ran into the people at Jaron. And I began to see the backbone of the elephant, where you begin to put together the ear and the tail and the trunk and the tusks and so on. So I think that's part of what we need to do. One is expand our horizons, not just talk about shortgevity, but real longevity, really change the world. Talk about transhumanism. I mean, not just extend the age a little bit, 5, 10 years. No, we're talking about hundreds of years, centuries, potentially. The second is, as I say, to realize that we have to understand aging. It is not simply a passive process of entropy. It's much more complicated than that. But the very fact that it's complicated means that there are multiple points of intervention, some of which are far better than others. And then I get into this whole discussion in the book that is a bother for me, because people focus so much on hallmarks and biomarkers, and hallmarks and biomarkers are not aging. They're critical measures of aging. It's like if I have a patient in the ICU with COVID, I will measure the oximetry, I will measure the leukocyte count, I will measure the temperature, I will measure their aches and pains, I will measure their C-reactive protein, but none of those are COVID. They are measures of what's going on clinically, but those aren't COVID. And if I want to cure a patient with COVID, I don't simply give them more oxygen. I don't simply say, I'll lower your temperature. I don't simply say, I'll change the inflammation. I'll put them on steroid. That doesn't cure the process. And the same is true of aging. Aging has lots of biomarkers. And as an example, when we go to our Alzheimer's trials, we'll be looking at a lot of biomarkers. For example, we'll be looking at a number of proteins that are involved with beta amyloid and C-react just anything we can think of. But those aren't Alzheimer's. Alzheimer's is not beta amyloid and it's not tau tangles. Those are outcomes of Alzheimer's. So we need to measure them, and they are critical in understanding whether we're making a difference or not. But biomarkers are not aging. Biomarkers are simply part of it, generally a downstream part of it. And the same is true of upstream parts. So if I'm looking, for example, at Alzheimer's disease, I know more than 200 genes that have a role. A classic is to be biallelic for ApoE4. But that's not Alzheimer's. Those are risk factors. So are head injuries, closed head injuries. So are a history of encephalitis. So are a history of exposure to a number of toxins, likewise radiation. All these things play a role as a risk factors upstream. And downstream, we have a lot of measures we can look at, biomarkers. But we need to look at the system, not just the upstream risk factors and the downstream biomarkers, but what happens. As I said in this conference, the Alzheimer's Association had me organized in Washington a few years ago. I was supposed to summarize what everybody said. I said, I'm not going to summarize what you said. You know what you said, but let me point something out. We've talked about the upstream risk factors. We've talked about the downstream biomarkers, but nobody has said, how do they go together? If I have a head injury, why does that increase my risk? If I'm biallelic for ApoE4, why does that increase my risk? Be specific. And what you find is that once you do that and look at it systemically, you no longer are talking about just points of intervention randomly. You're asking yourself, what's the optimal point of intervention, both clinically and financially, which is a critical question. Now, we're picking Alzheimer's because it's the best strategic target. It's fatal, and more than 3,245 interventional trials as of yesterday aside, there is no cure for Alzheimer's. You statistically can slow it down with Leqembi and aducanumab, but that's not the same as stopping it, let alone reversing it. So it's a fatal disease with no treatment. So for us, it's a no-brainer. I'd rather do that than going after osteoporosis, for example, which tends not to be a fatal disease. It can increase your risk of falling. People go into the hospital, they get an infection, they get your sepsis, they end up in the ICU and they die, but they don't die of osteoporosis. So Alzheimer's is different. It scares all of us, and it should. It's the leading cause of death in the UK this year. In the US, it's not the leading cause, but it depends how you define it. No, it's our strategic target, but it's a systems issue. It's not biomarkers and it's not risk factors. It's the whole thing as a system. So that's kind of what got me into doing the book. And then I wanted to point out that if we do this, it'll have dramatic consequences for us culturally, socially, ethically.
Yes, thank you very much, Michael, for that overview. And I completely agree with you, of course, that what we should be pursuing is radical life extension, age reversal, living hundreds, thousands of years longer, potentially indefinitely, not just trying to get a few more years of life. And the Transhumanist Party seeks to catalyze a change in the mindset surrounding aging and what can be done to address it and hopefully reverse it. And we have David Wood joining us. Welcome, David. We just heard an overview from Michael Fossel of essentially the key framework of thinking that motivated his book. Again, here is the cover of the book, Reversal, Science, Medicine, and a Future Beyond Aging. And you can see the two backward arrows in place of the V, a neat little element of the graphic design of this cover. We're getting some good audience feedback. Our friend Mike Lazine writes, I'm really liking Dr. Fossel. So thank you very much to Mike for joining us today. And there is a question that Mike Lazine has. He wonders, do you think, Dr. Fossel, that to live hundreds of years, we would need to hack our DNA and RNA at a microscopic level? Or could some other approach work? If the problem is telomere shortening, would we need to do gene editing, or could something else, some other more macro level approach suffice at reversing the shortening of telomeres?
I always find the verb hacking a little odd because it means different things to different people. It's a great verb. It just, I say hacking, you say hacking, and we're thinking about different things. But yes, I think we can hack things. But in my mind, the question isn't sort of what causes aging. I find that a lazy question. It's like, what caused us to be on this program today? Well, what are you really asking? What do you mean, what caused us to be here today? And I look at it in a more practical standpoint. I think the question of what causes aging is interesting. I should be discussing it not over a cup of coffee, but over a glass of wine. But a practical question for me as a physician is not what causes it, but what's the optimal point of intervention? And it bears on your question, do we go after the DNA, the RNA, the biomarkers, the upstream risk factors? And I still think that to use an analogy of the symphony orchestra might help us here. And this will, I think, resonate with Bill too. Let's imagine a symphony orchestra, and each of the instruments is a gene, and each of the musicians is the epigenetic control of the gene. And Bill likened this once to a dimmer switch. All right, it's even more complicated than a dimmer switch because, again, I'm the first violin. It's not just how loudly I play, but exactly what notes I play in response to other notes. It's very complicated. So let's imagine a symphony orchestra. And the symphony orchestra, as we age, I have the same genes at age 7 that I have now at age 75, almost the same genes. There are some changes here and there, but basically the question is not the change in genes, it's the change in gene expression. That is, if I look at the symphony orchestra of me when I was 7, it may have been playing Mozart, and now it's playing some atonal John Cage piece. Nothing against John Cage, not my thing, but the point is it's playing a different tune. It's the same first violin, and the first violinist is not hungover. She's fine. The piano's in tune. The pianist is still well-trained from Juilliard. The orchestra is the same at age 7 and age 70, 75, almost, okay? The difference is the tune they're playing. It has to do with the orchestral leader, the maestro, the score that they've chosen. And if I wanted to say that I want that orchestra to go back and play the tune it played when I was 7, I could go around to each and every musician and say, listen, never mind what you're playing, play this little piece, play this little piece. That's equivalent to me trying to deal with methylation or Yamanaka factors. I could go after the string section as opposed to the percussion section, as opposed to the brass section. What I'm suggesting is that the most efficient, the optimal point of intervention, as I say, clinically and financially, the efficient point is to go to the orchestral leader and say, no, skip that score, play this one instead. And what we find is that it actually works. I mean, Bill knows this. The first article came out in 1998 looking at doing this in fibroblasts and human fibroblasts, saying you can reset the orchestral score. And then in human tissues in 2000 and 2001, both in subendothelial cells, vascular cells, and in bone cells and so forth. And we've seen that since in animals too. Now, the question isn't, does it work? It's how good are we at replacing the score? And we are fair at that. We're a lot better now than we were in 2001 when I got offered tons of money to do this because we didn't have the techniques we needed, let alone the genetic techniques and the problem with gene therapy then. We're getting very close. So yes, I think we can go in and replace the score. And it's not that telomeres or the score cause the orchestra, cause aging. It's just part of the enormously complex systemic process. And it's the most efficient point of intervention because it works. So yes, I think we can hack things at the DNA and RNA level. And that brings up some specific issues too, which is how do you do it? Do you use, for example, an AAV virus or a lentivirus or something similar? And if so, what serotype and what promoter and exactly which gene? And do you alter that slightly? Do you go the other extreme and use lipid nanoparticles and mRNA? And the answer is that entire technical process is changing so fast, it's hard to keep up. It changes literally week to week. So we're going to be looking at both in our large animal studies probably next year. But it's a good question. Where do you hack it? I don't know. We'll find out.
Yes, thank you very much, Michael. And I'm curious, Bill, since Michael addressed some of your thinking in his comments, what do you think in response to his statements just now?
Are you asking me? Okay, so
yes.
I thought that the orchestra leader thing was an absolutely perfect analogy. I when I was still at Geron Corporation, I remember being in Calvin Harley's office and drawing on the whiteboard in his office about how telomeres have all the hallmarks of enhancer sequences, and that its structure function suggests that there's no way it couldn't be an enhancer sequence given everything, and that the telomeres are actually the orchestra leader that are controlling the expression of the genes. And it's I can explain that a lot more because people ask, well, how does it control genes that are a long ways away? But it just has to control the transcription factors that are near the telomeres. So I think that we're going to see some dramatic results, especially from like Mike's clinical studies that he's doing, that not only is he going to see Alzheimer's get cured, I hope, he's going to see some people show really signs of obvious age reversal. I do agree with something he said earlier about the biomarkers. Yeah, biomarkers, they just correlate with aging. Reversing a biomarker doesn't mean you reverse your aging. It just means you reverse the biomarker. So I don't know. Mike was actually one of my mentors when I first got into the field. He's one of the few people that has been in this field longer than I have. And so I actually got to learn a lot about telomerase and telomeres from Mike. Even though I was at Geron Corporation, I was having a hard time learning it there from everybody else. But yeah, everything Mike just said, I agree with totally.
Bill did all the work. I was just the one with my hands on patients. That's all. But Bill was the actual one who knew more than I did by a long way. Yeah. Always asking.
But you were obsessed with we need to cure aging, and so was I. And there were a lot of people at Geron that just weren't. So, yeah.
Yes, indeed. Well, I'm glad to learn a bit more about the background of the acquaintance between Bill and Michael and your history of collaborations. Also, we have a question from David Wood. He says, neurons don't divide as far as I know. They remain in place throughout someone's life. So how can lengthening telomeres help with brain aging? That's an interesting question. And Michael, if you have any thoughts on that, I would be curious. And Bill, if you have anything to add after that as well, please go ahead. But Michael, what thoughts do you have on brain aging and telomere lengthening?
A couple of things. One is that, the traditional teaching, I used to teach this at Stanford, and that was that neurons don't divide in the adult. Well, mostly they don't, but they clearly do. We see some of this in the limbic system. We see some of the cerebellum. We see some of it here and there. We also see the same thing in animals. And in fact, the nice study that was done in mice years ago found that you can actually, if you reset telomere lengths in mice, you actually get increase in neural stem cell differentiation and regrowth of brain volume. So in general, I still think you're right, David. In general, most neurons don't divide. And even if they did, remember that the key to a neuron is not the key to my liver cells or how many liver cells do I have, but the key to a neuron is not how many do you have, but the precise connections. And I don't just mean the synapses, but again, the relationships between those synapses and how they fire relationship to other synapses near, it's just tremendously complicated. So you can't just say, well, I got a dead neuron, put another one in. That would be like saying, I lost my CPU, put a new CPU in, and what happened to the memory? Where's it gone? It is important. So yes, neurons somewhat divide, and that doesn't necessarily mean it's going to solve anything if you can replace them, because it's the connections that matter. Also, we know that the astrocytes and the microglia are tremendously important as well. But even then, it's true that they're more likely to divide, particularly microglia. But again, there are structural constraints. The relationship between that microglia and that neuron and sort of the Fourier transform, the interactive effects are critical. So you can't just say, well, we're going to randomly replace 500 microglia. That's nice. And yet it seems to make a difference when we do that in mice. So, but there are limits. The other thing that comes up is we know that in non-dividing neurons, we do see age-related changes, even though theoretically the telomere hasn't shortened, although there is some interesting side data on that I'll ignore. But if I'm a neuron, it's not just a question of what my gene expression would be if my telomeres didn't change, but what's going on with all the astrocytes and microglia nearby. And if they're showing senescent changes, they're going to put a tremendous stress on not only my cell function, but changes in my epigenetic pattern. So things get complicated. And there's no way I can pin down precise answers on this because with just so much we don't know yet. But again, if we look at what happens when you reset telomere lengths in mice, either by doing it in the germ cell lineage or by doing it in adult animals, we see tremendous changes not only in the slides that I've seen in Maria Blasco's work in Madrid, but in the actual behavioral changes in the mice involved. So what's interesting about that is, for example, in Maria Blasco's work, she was only transfecting maybe 1% of the neurons and maybe 5% of the microglia. We hopefully can do a lot better than that. But even at that level, you found better behavior in animals. So the question is not just can we transfect neurons and can we replace neurons, but what are the overall effects if we're really good at it? And here's another part of that. Let's say I'm making up these numbers, all right? Let's say I've got a patient with advanced Alzheimer's and that 90% of the neurons are fine, 9% are pretty sick, and 1% are dead. I made that up, okay? I can't get back the dead neurons. And as I just said, if I can get them back, what does that do to the interactions, the memory, the behavioral data that's stored in those interactive effects? However, I can probably improve the function of the 9% that are merely sick. So I think there's a lot of room for us to increase function in patients with Alzheimer's. Having said that, I still think there are what I used to call Humpty Dumpty effects. Some things are broken and you can't fix them. If I spill coffee, as I've done before, I don't know if you've ever done this, on my laptop, and I take it into somebody to get it fixed, I don't expect it's going to be perfect. The question is, how much function can I get back? I've had to replace the keyboard twice because I'm such a klutz. But the same thing is really true of Alzheimer's patients. I think that we can't, there are many scales for Alzheimer's, clinical scales for Alzheimer's. Let's take a typical one, 0 to 6. Hopefully most of us are zeros. We're doing fine. 6 is that patient the last few months in the nursing home, they are fed, their bed plans change, they don't recognize that they're alive, and they're not breathing very well, okay? If I have a patient who's a 6, I'm not going to get them back very far. If I've got a patient who a 1, I can probably get them to a 0. We're looking for patients who are 3s and 4s, partly because if it ain't broke, I can't fix it, partly because we're doing something that people shouldn't believe we can do. As I said, there have been more than 3,245 interventional trials for Alzheimer's. By global consensus, they haven't cured Alzheimer's. So if I come along and have data that suggests I can, no one in their right mind should believe this. So we're concerned mostly about credibility as well as safety, efficacy, cost, and timing. But credibility is a big deal. So we're looking for patients who clearly have Alzheimer's. You can't say, nah, they really didn't have it, so you didn't cure it. They've got it. And yet I recognize the message behind what David said, which is some things are broke, I can't fix them.
Yes, thank you very much, Michael, for that answer. And Bill, do you have any thoughts on the question of neurons and the relationship of telomeres to the aging of neurons?
Just almost repeating just what Mike said, but neurons, yeah, well, even though they don't divide, that doesn't mean necessarily that telomeres can't be lengthened in them. They don't shorten by what's called the end replication problem when cells divide, but they could still shorten from oxidative stress problems, things like that. And we don't really know if we can lengthen telomeres in non-dividing cells. But the key is that there is the microglia and the astrocytes. And the main microglia I usually talk about are the oligodendrocytes that are making the myelin that wrap around the neurons. I look at those as, I call them all the time, the bodyguard cells and the caregiver cells, okay? Because they're the ones that take care of the neurons. Neurons are the healthiest, longest living cells in the entire body. They can clearly outlive any of us now. But it's like when the neurons, the microglia, astrocytes, all the glia cells, and oligodendrocytes, and even in the peripheral nervous system with the Schwann cells, they are all having telomere shortening because they are all dividing. And so keeping the telomeres long in those cells might be the best way to be keeping the health of the neurons going. Let's see, and there was anything else I wanted to say. I was quite surprised with Rhonda Pinnell's mouse experiment,
right,
where brain functions came back.
Right.
Mike, did you want to add anything about Rhonda Pinnell's stuff?
Yeah, because one of the things that fascinated me about that, I mean, what he did was he used an estrogen switch on the germ cell lineages. So it would sort of be like me saying, I can't cure your Alzheimer's, but your great-great-great-granddaughter won't have it. Okay, that's good. But what about me? What he found is that in the aged mice, they had lost about 75% of brain volume. And after he treated them by turning on the estrogen switch, it went up to 89.7% of brain volume. And they had behavior improvements. So, and what he found was when you looked, you found these neural stem cells, and as I say, increased differentiation. So clearly there's a lot we don't know. Clearly there's good potential for being able to intervene effectively in a clinical setting. The question is, how do we do it, and how far can we go with this? Give the other piece of this, the exceptions. As I say, some neurons are dead. You know, the longest neuron in your body is about 1 meter long. You take a BET cell that goes from the brain down to the bottom of your spinal cord, say down the sacrum. And that motor neuron, if we lose that, it's not clear how you ever get it back. Let me give you an analogy of how specific it is. That neuron, by size and by distance that it travels and specificity, is like this. It would be like me sitting in Times Square, reaching out my arm 3,000 miles across the continent to a specific person in Los Angeles by the town hall, and not the person sitting next to him, and setting up a contact. That's how specific those are. So if I lose that neuron, what are the odds of me getting it back? Again, Humpty Dumpty effects. We don't know, but you see the task involved, or the difficulty involved. And yet you see the data that Rhonda Pinnell got when he was at Harvard before he went off to Texas, or the data that Maria Blasco gets. It's impressive that even with our limitations, we get that good a result. So let's see what we can do in human trials.
Yes, thank you very much, Michael and Bill, for your responses on this. And we will have a few follow-up questions about neurons and brain aging, but first, a bit more of a general comment from Daniel Tweed, our Director of Community and Citizen Science. He says he likes the orchestra model of aging, and he observes the orchestra conductor and the orchestra are aging at the same rate after all. And well, this is the case for human orchestras right now, but I think what he's hinting at is whatever that conductor mechanism or process is within the human body is itself aging and deteriorating. So if it is telomere maintenance, essentially, that is the conductor, then telomere shortening is that aging of the conductor. Now, Daniel does wonder in regard to a particular creature, the octopus, which has an average lifespan of about 3 years, but apparently octopi are able to hack their own epigenetic code. So, Michael, since you've had some involvement in animal studies, I'm curious if you're aware of what happens with octopi and their aging and why they have such short lifespans, given that their ability of epigenetic editing is something remarkable that we humans don't seem to have.
Let me preface this with a humorous comment. Back in that Alzheimer's conference I helped organize in Washington on animal models, I got up and I said, I just realized that whether you're looking at mice, rats, cats, dogs, rhesus monkeys, gorillas, or humans, and there's a species or two I'm missing, in my clinical and research life, I have been bitten by all of these. And when I say humans, I don't just mean the kids either. I've never been bitten by an octopus, but I have been bitten by a gorilla, by the way. And as I say, rhesus monkeys, I still have the scars. I hate rhesus monkeys. Gorillas are nice people. I avoided chimps, thank God. But I don't know about octopi. When you say they can hack their epigenetic code, I sort of wonder what that means, because we hack our own epigenetic code. You can hack it by your diet and whether you smoke or drink and a million things. So a little unclear. But back to Bill's a point that he made once recently about lifespan. Let me point out that, if I'm, here's an example. Let's take two islands. And on one island, I've got deer that live for 500 years. And the other, I've got deer that only live 1 year. And they're both have good populations of deer. Slightly different species, but otherwise, lifespan's the only real difference. Now I change something. I change the pH of the water. I change the temperature. I bring in a new a predator. I move some prey. I change the vegetation. Anything I do, the ones that only live 1 year will be more responsive. They have a shorter turning radius than the ones that live 500 years. So there are many advantages, I think, to living a long lifespan. But on the other hand, when the meteor hit 66 million years ago, the dinosaurs didn't do very well. It was the little teeny critters who managed to survive and adapt faster turning radius. So what I suspect is going on with not just octopi, but all the rest of us, is that there's a very finely tuned mechanism. Not only is it a balance between long and short lifespans and survival of the species, because it's not just your survival, it's the whole species, but that is responsive to changes in the environment. So for example, if I have a stressful environment in a teleologic sense, I'd like a shorter lifespan so that I can adapt faster. I'd also like a higher mutation rate. Well, when you shorten the telomeres, guess what happens? Not only do you have a shorter lifespan, but you have a higher mutation rate. DNA repair slows down. The turnover rate, the recycling rate for the 4 major families of DNA repair enzymes slow down. So I find it fascinating that anytime you induce increased stress, you get more cell turnover, you get shorter telomeres, you get more mutations. That's not a good idea for you personally, but from a species perspective, you don't want a lot of mutations, but you do want some. If you have no mutations, nothing adapts. If you have too many, the species dies out anyway. So not only do you want mutations, but you want it to be responsive to changes in the environment. And I'm suspecting that that's part of the mechanism here. I don't know. This is more philosophy than biology, but I just suspect that that's part of what's going on with regard to telomeres and epigenetic changes. Yeah, you noticed, if you listen carefully, I didn't at all answer the question about octopi. Sorry about that.
Hey,
thank you.
Yeah,
I have been bitten by an octopus.
Really?
I used to keep pets octopus, and I'm not a semantics expert because of my experience with octopus. The plural of octopus is not octopi; it's octopuses, it's irrelevant. But they have a beak right in the middle between their legs. A beak that actually bites, and if you let the one on your arm wrap around your arm, you can get bitten, and I've been bitten. But I can't remember what else I was going to say, but it was nothing important.
Octopi and epigenetic changes. I don't know, hacking them.
Well, it's interesting. We're learning as well that for whatever reason, telomere biologists seem to have a greater than ordinary statistical propensity to be bitten by various creatures. And that is a curious question to consider at some point, perhaps because of greater exposure to a variety of animals. But it is also fascinating to consider the differences in aging mechanisms and rates of aging among animals. And Michael, to your point, and I thought this was an excellent observation, the stress in the environment that a species typically faces will affect the rate of aging and the length of typical lifespans. And the less stressful an environment is, the longer the lifespans that are evolutionarily selected for. This also, interestingly enough, sheds light as to why organizations like the Transhumanist Party have generally been advocating for a calmer world. We don't like the turbulence that is happening in the world right now, all of the geopolitical tensions, the societal upheavals, because that turbulence creates stress, and that stress is bad for individuals. It may lead to some societal adaptations, faster turnover, but faster turnover of people is not a good outcome in our view if we want to lengthen lifespans. So calmer periods of history, greater societal stability, greater certainty of background conditions that people encounter are conducive to human longevity, it would seem to me. And we do have a question along these lines from Kevin Bransaforti, and he wonders, what lifestyle habits can people adopt to help support their orchestra or score to maintain cellular health until more longevity technology is achieved? So our mutual friend, Dr. Aubrey de Grey, has given a probabilistic estimate. He thinks that there is about a 50% probability that with adequate funding, reversal of aging, the attainment of longevity escape velocity can be reached within about 10 to 15 years. So sometime in the late 2030s. But we need to keep ourselves alive and in good health before then. And this is where lifestyle does matter. Now, I know a lot about what Bill Andrews does in order to support his organism to prevent some of the deleterious consequences of aging, because we had a 3-hour virtual Enlightenment Salon with Bill on October 10th, 2021. And he talked as part of that salon about his running habits, about his approach to nutrition, to supplementation. And Bill is somebody who takes this extremely seriously. He is one of the most rigorous people I know in regard to focusing on ways that aging can be slowed at present. And Michael, it seems that you've had some significant success in this as well. So both Bill and Michael were born in the very early 1950s, and neither of you look like somebody who was born in the early 1950s, at least having seen a lot of people in their mid-70s. You two are outliers. So Michael, I'm wondering, what do you think can be done today in order to help tide people over, create that bridge so that if any aspect of aging can be slowed down today to help people make it to the late 2030s, they could pursue that?
Yeah, you're right. I was born in 1950. In the book, I've got 2 chapters, there are a lot of chapters, 10 chapters, but there are 2 chapters that bear on this. One chapter said, what can you do? And the other chapter is, what can we do? And the what can we do is what we've been talking about, resetting telomere links, reversing aging. But the question is, what can you do? That is, how can you slow aging? And in a facetious sense, the answer is you should be doing what your grandmother and your doctor told you, and you didn't pay attention to either one of them. And your doctor costs more than your grandmother does. But what I'm getting at is I think most of us are aware of what we should be doing. You should eat right, you should exercise, you should lower your stress, you shouldn't smoke, you should fasten your seatbelt. That'll extend your lifespan. It doesn't change aging, but there are a lot of non-sexy things we'd all should be doing. But one, as I say, that doesn't stop aging. But yes, you can slow it by being, in a sense, reasonable, I think is one way to put it. But even then, you have to be careful, quote, how reasonable you are in a sense. Bill runs into this as an ultramarathoner. And the question is it better for a system to run the ultramarathon, or is it worse for his knees, for example? And that's a real question. And it's one that Bill thinks about, and he comes up with the answer to that. We know that you can over-exercise and damage yourself. You can under-exercise and damage yourself. So what's the balance? The same thing is true with regard to diet, for example. I used to do this course on the biology of aging, and I talk about what's the optimal diet. And the first thing I'd do in front of the class is I'd put up a picture of a very small mammal. And I'd say, if any of you can identify this mammal, you'll get 5 points in your final grade. Absolutely. Nobody could do it. It was Lemus, the lemming. Now, lemmings don't actually rush off cliffs, but it is a typical lemming behavior that we all have when it comes to diet, for example. So you'll find that this is the diet that make you live forever. This is the diet, and people will argue: high carb, low carb, high protein, low protein, high fat, low fat. And the very fact that people get so caught up in this suggests to me that, one, we don't know as much as we should, and two, people typically like to run off cliffs. It's the diet of the week, the diet of the month, the diet of the year. But I've seen the same thing happen with regard to things that shouldn't be that way at all. Example, stress. I've had several times where people come in to see me in the hospital and say, I've got a problem. My blood pressure is too high. I said, how do you know that? I've checked it 10 times today. Well, the first thing you need to do is take away your blood pressure cuff. Stop that. You know, the very fact that you're so focused on trying to deal with my stress is stressful. And the same thing happens with diet. People get so concerned about diet or so concerned about exercise, they lose touch with the reality of it. So again, back to Bill and ultramarathons. I don't think Bill or I would recommend that everybody do ultramarathons, but it may be the perfect thing for him. I don't recommend that people should sit on the couch either. And yet sometimes the answer is, yeah, just chill for a while. So back to, what should you be doing? Yes, a lot of the things we know. When you come to issues like a supplementation, you run into the same problem. If you look at it in a broader historical perspective, you begin to realize that what we thought last year is not the thing we think this year. Did the reality change, or is it just our perceptions of it? I guess what I'm saying is, and I put this way on purpose, is with regard to diet, we should take it all with a grain of salt.
Yes, thank you, Michael, for that response. And taking diet with a grain of salt, that's quite a nice turn of phrase. Now, Daniel Tweed observes checking your blood pressure.
Can I make a comment?
Yes, go ahead.
Really quick. So I agree totally with the kind of Goldilocks effects that Mike was just talking about, but overdoing it, underdoing it, and stuff like that. But it's like I just say, keep it fun and keep it consistent.
Yes,
and, that's the secret. And I had the benefit of having an identical twin brother who does not run, who does not exercise, and he can barely walk. He's got terrible knee problems, hip problems, ankle problems, and I think it's because of not exercising enough. But I think that moving, always being consistent, running all the time, but keeping it fun is a great way to keep joints and hips and things like that lubricated. And as a result, you don't have knee. I don't have any knee problems. I've been running since I was 2 years old. Okay, I've competed in high school cross country, track, also in college. I did the same thing. I've been running ultra marathons. I used to hold the world record for the most 100-mile races run in a year. I have no joint problems at all. And it's not genetics, it's lifestyle.
Yeah, it's funny when you mention joints, because one of the things that fascinates me about joints anatomically, physiologically, and medically is that, I'm going to, again, simplify. The chondrocytes in your joints have no blood supply. It's all passive, because if you had a blood supply, it'd bleed and your knee would be full of blood. Again, teleology. But what you find is that the chondrocytes typically are like sponges. If I have a sponge full of paint and I'm trying to clean it out, I squeeze it under the water, I release it, I squeeze it under the water, I hope it's latex paint. Squeeze it, release it, and after a while, I've got water instead of latex paint. But it's the same thing with your chondrocytes. The way that they get oxygen, the way that they get sugars, the way that they get vitamins and micronutrients, the way they get rid of carbon dioxide and waste products is by being squeezed and released, squeezed and released. And every joint, in a sense, has a native rhythm to it. You are not meant to sit quietly and not move, or your joints don't do well. Likewise, you're not meant to take a sledgehammer to them. But there are reasonable ways of treating your joints, and it requires a certain amount of, as Bill says, motion, daily motions.
And I learned about chondrocytes from listening to Michael talk in the early 1990s. So,
Yes. Well, thank you for that discussion. And Miriam Abbas writes, that's a good analogy, Michael. she also has an interesting question. She wonders, how can we live like the immortal sea creatures? And I think she's referring to the immortal jellyfish, the Turritopsis dornii, which can differentiate back into its infant state. Is there any hope that there's any sort of common age reversal mechanism between these jellyfish and what humans can potentially apply to themselves?
Conceptually, yes. In a totally practical medical sense, only distantly. Back to what Bill and I have been talking about for more than 3 decades. If we can reset telomere lengths, we can probably have the conceptual analogy of the immortal sea creature. Again, within limits. Here's a thing that doesn't apply. People tend to assume that, as I say, aging just happens. Well, there are some non-aging changes that occur. Example: if I have normal lungs, I can get idiopathic pulmonary fibrosis. Just think of it as old lungs. Okay. If I can reset telomere lengths, I can probably reverse idiopathic pulmonary fibrosis. But what if I'm a miner, and over the past 30 years, I've gotten silicon deposits in my lungs, silicosis? That's not aging, but it sure creates disasters as you get older. And I can't, no matter how young I make your lungs, that doesn't pull the silicon out. It may remove the inflammatory changes. It may give you perfectly normal pneumocytes, but it doesn't take out the silicon load you put in there over 30 years. So what I'm saying is that, yeah, in principle, we can probably have you act just like the jellyfish, the immortal jellyfish. But it sort of, again, depends what you do. If you're a miner, you're going to have old lungs, and it has nothing to do with aging. It has to do with accumulation.
I want to add to that.
Please.
You think about culture of bacteria growing in petri dishes. They'll keep growing and growing and growing and growing. I like to think of jellyfish as actually a culture of bacteria, except they're all joined together, okay, into one organism. Because that's why you can cut a jellyfish in half, and the 2 halves will grow into a new jellyfish. You can actually dissociate the jellyfish into single cells, and those will grow into jellyfish.
We're going to try cutting Bill and I in half next year and see what happens to us.
Yeah, right. But it's like I always say, even the trees that live forever seem to live forever. Sometimes they only have like one branch at the very top of the tree that's still alive. The rest of the tree is dead. And it's like, I don't want to be considered alive if my little finger is still alive, but the rest of my body's dead. So humans are very different than jellyfish and trees and all these other animals you always hear about that have these long, extensive, immortal lifespans. They're just not human.
So again, to make the same point, to reinforce what Bill is saying, conceptually, yes. Practically, we're not jellyfish.
Yes, fair enough. Though I would say Stephen Hawking went a long way with just being able to twitch one side of his lip because he had a computer connected to himself, essentially, which was able to interpret those twitches, and he could type, essentially. It took him longer than most people to respond to inquiries, but his brain was still functional. It's just his body was severely paralyzed. But I've joked that if I were disabled in that manner in the future, where I could maybe only move one finger or so, I would probably connect my finger to a large language model that is based on essentially my writings and other information about me, and I would still try to respond to events as they occur. So I would have input into that system that I'm connected to regarding, say, current events, or if somebody wants my thoughts on something, and if I move my finger this way, one type of output would appear, and if I move my finger that way, a different type of output would appear. So I would still somehow be acting upon that system and trying to exert my influence upon the world. Obviously not an ideal outcome, but I like to see myself as a survivor, and I would try to survive in those circumstances. We have a great
Hawkins' brain is a lot more important than my little finger. So
actually, if we can, let me take this down an AI road temporarily too. Sure. I don't know about the rest of you, but I can't get through a day without an AI. I have 7 of them on my cell phone alone, and I'm using at least 2 every day on my laptop. But one of the comments I want to, two comments I want to make. One is that we intend to not only have a clinical mode, but use AI to help us do a lot of things that we're going to do to take this to the world, essentially. I mean, we want to make sure it's not just something that it costs you a lot of money to afford, but everybody can afford it. But that involves AIs. The other comment I'd like to make, and I made it in the book about the future of AI in medicine, is, the old GIGO, garbage in, garbage out. The same thing is true, I find, with regard to using AIs, which is quality in, quality out. I'll give you an example. If I'm going to use an AI in a very naive sense medically, I would say, Gennady, you've got 7 lab tests. Let's give this to the AI. AI, what has he got? Wrong question. Okay. Here's a slightly better question. I could turn to the AI and say, listen, here's 7 lab tests from him. I want you to construct a differential diagnosis of the first 99% probabilities. List them all in order of probability. This one's 60%, this one's 39%, this one's 12%. List them all and tell why you think that, and tell what tests I can do to rule in, rule out, or at least raise or lower the probabilities of each of those differential diagnoses. Slightly better example, okay? But I really should be saying something a little more complicated, which is, it's not a matter of the diagnosis. What about interactive effects, for example? If I'm using, there's a drug, an ophthalmic drug called Simbrinza that you can use for if you've got high pressure in your eye, but it causes a rash. Now, if somebody has a rash, but it itches and it doesn't go away very fast, is it because they secondarily have an invasive dermatophyte? That is, they've got a fungal infection around the eye? If I just said what caused it, wrong question. I should be saying more complexly what kinds of things. And I've seen this in patients where a woman I know fainted essentially while she was on a treadmill. And what I found is that the neurologist had one answer, the cardiologist had another answer, the family practice had another answer, and none of them put things together. So back to AIs and what does it do to longevity, for example, and medicine in general, it sort of depends on how stupid we are in the way we ask questions. I find people are worried that we'll get so used to AIs that we stop thinking. I find that AIs force me to think more carefully and think about my prompts to get the right answer. Again, quality in, quality out.
Absolutely. And the prompting is essential. I think the human element in interacting with an AI and understanding what uses it's capable of being put to right now and what areas it's still limited in, all of that is a key human contribution that will not be made obsolete or redundant anytime soon. We have a wonderful comment and then a question from Katie Clark. She says, thank you, Gennady, the Transhumanist Party, and to all our guests for bringing these conversations to the public. Keep leading the way in transparency of knowledge for all. And thank you, Katie, for your generous support via this super chat. And she has a question for Michael. She writes, Dr. Fossel, do you agree that sustained AI partnership creates new cognitive pathways through neuroplasticity, allowing in a degrading brain, for instance, a brain with Alzheimer's, dementia, TBI, to route around damaged segments? So I know you're planning to conduct a study on Alzheimer's patients. Do you think there is a potential application of AI to help them stave off, if not reverse, the progression of Alzheimer's disease?
I would answer that in 3 ways. The first is back to the comment that was already made that you and Bill have talked about. If I can twitch one finger, well, I can use AI to take that twitch and do more than I could have otherwise. Okay, that's one example. The other, far down the, or a little further down the road example would be, what if I can do an implant? What if I can essentially have a really relevant quantum computer with AI function in my brain? That's certainly a possibility. Not today, but we're getting closer. But in between that is the question that I think that Katie's asking right now, which is, will the AI itself, my interaction with an LLM or something like that, will that change my plasticity? Yes, I think it will, but not sufficiently. That would be my quick answer. I think that we'd be much better off to reset telomeres and bring back your brain function.
Yes. So the change in plasticity from the use of AI would not be sufficient to reverse or significantly halt the progression of Alzheimer's disease, but perhaps essentially reversing the shortening of telomeres could have a greater effect. So thank you very much for that response. And David Wood has a suggestion in terms of the use of AI. Once the salon is over, he suggests people ask their favorite AI models, can octopuses alter their own DNA? ChatGPT has given a lot of fascinating info about A to I RNA editing and jumping genes. So thank you for that remark, David. And this is another excellent use of AI systems, especially if one puts the same question to multiple AI systems. That way, any sort of hallucination or strange interpretation of the question would be checked by the other models. And Michael, you said you use about 7 models on a regular basis. Do you find it useful to have them essentially cross-check one another? And how do you address divergent interpretations among the different AI models? Do you have any sort of heuristic that you follow if you get different answers from different models in substantive ways?
Well, first, as I think all of us know, they have different specialties, different abilities. I mean, Art must have run into this in dealing with art. With dealing with graphics, art. I could ask Gemini, I could ask ChatGPT, I could ask, a whole hassle of them to come up with a graphic for me. Some of them can't do it, and they'll all come up with different sort of interpretations. And after a while, you have to ask yourself, which one is most helpful to me? And this is true of anything I do. So back to the issue of, are they reliable? When I was writing the section about age-related disease, and I got to the section on bones, one of the things I like to do is have a little what's called an epigraph at the beginning of each section, a little poem, a quote from a Chinese philosopher, a quote from the Edda, anything, a little that bears on that issue. So when it came to osteoarthritis or osteoporosis, I thought, I need a poem that has something to do with bones. I couldn't think of one. So I asked, who did I ask? It wasn't Gemini, one of the other ones. I asked, come up with 6 poems that talk about bones. It did. So here they are. I looked through, nothing about bones at all. I said, there's nothing about bones. I said, oh, I'm so sorry about that. You're right. I made a mistake. Here are 6 that do. Nothing about bones. After a while, I gave up on that because I realized it wasn't going to work. So you know that there are limits, and it depends which one you ask and what you're asking, how you ask it. But let me put that in an even broader perspective. It depends on how narrow your assumptions are. So example, let's say that we get in a time machine and we go back to 1895 in Paris. And one of the rages is hot air balloons. And one of the problems was, how do we design a really good hot air balloon in terms of the actual cloth structure, in terms of the heating element? And a big problem back then was, how do you land without killing you, without crashing? Okay, important questions. So you and I go back in our time machine, and we go back with Gemini, and we go back with ChatGPT, and we go back with 3 or 4 others, and a quantum computer, and anything else you want. And we say, go ahead, now you can really do something. And what they'll do is they'll ask you to design the perfect hot air balloon. Wrong question. They're assuming you can't, back then, Lord Kelvin, the world's preeminent physicist, said we'll never have heavier than air flight because it's impossible. Okay. So they were asking the wrong question. And no matter how good the computer had been, quantum computers, 7 AIs, they were still asking the wrong question. It was the wrong prompt because they were making assumptions that weren't valid. If I'd given it to the Wright brothers, they would have said, how do we actually design the engine so it's cheap and small, so it doesn't weigh much? How do we design the actual structure of the wires and the cotton cloth? Different question. So a lot of times when we're trying to make advances, for example, with aging, people will say, what's the right protein to use? Well, you've already asked the wrong question. And the problem is not that the computers aren't good. They're not perfect. They hallucinate. They give me the wrong answer about bones. The problem is that I'm not thinking about the way I should be thinking. I'm making assumptions that are unexamined. And that's why we end up with problems and people saying that, computers can't make intuitive jumps. Well, a lot of the problem is in our heads. We don't make intuitive jumps. We ask how to perfect the hot air balloon. Or if it was 1950, we'd be saying, how do I have the perfect iron lung for polio victims? That's the wrong question.
Yes, indeed. So being able to ask the right questions is a key human skill. And Mike Lezine notes AIs are only as intelligent as the human asking the AI the questions. So thank you.
Unintelligent.
Thank you, Mike, for pointing that out. And we do have a question from David Wood that I think would be good to ask at this stage. Dr. Fossel, can you say more about the large animal studies that you will be overseeing next year in terms of what animals would be involved, what you would be testing them for, what would be the hoped for outcomes as well?
Yeah, let me broaden the perspective on it. Back with this conference that we were doing on Alzheimer's animal models, one of the memes that everybody knew about was that, quote, everything works in mice and nothing works in humans, which is not at all true because most things don't work in mice no matter what you do either. But even if they work in mice, the problem is that, for example, with regard to beta amyloid, the amyloid physiology is totally different in a mouse than it is in us. And even if you take human genes and insert them in a transgenic mouse, you haven't inserted all the other genes that play a role in it. So even if you get a good result, it doesn't really work well when you take it to humans, and it's very frustrating. So the question of which animal to use has two aspects. The first is, well, what will teach you about humans? If I can do it in mice, but that, I could do it in jellyfish. What does that tell me about humans who's going to respond? The other one is a political issue. I'm going to say political. It's perhaps not fair, but over the past 30 years, the FDA requirements for a number of human trials have changed. They've shifted over time. And over the past 12 months, they've certainly shifted as well. And over the next 6 months, they'll probably shift again. We have a fondness in our executive committees to saying, our politics are simple. We're in favor of getting rid of Alzheimer's. That's it. That's our politics. And the same is really true when it comes to human trials. If the FDA would say to me, you need rhesus monkeys, much as I hate rhesus monkeys, we'd use rhesus monkeys. If they said that you needed to use lawyers, never mind that. If we had to, my wife's a lawyer, I have to be careful. If they said we could do it with mice, we do it with mice. If they said we could do it in silico, we do it in silico. And our concern is not so much what we're required to do as just tell us, and we'll do that. Now, the default currently is probably to do it in dogs. I've got 3 dogs. I love dogs, probably beagles. I've had a beagle. They're the dumbest breed I've ever had. But I love beagles. But what we'd be doing if we do it in beagles is older beagles and trying to show that we can actually reverse the cognitive decline in beagles, as well as a number of other measures, biomarkers, for example, cardiovascular measures, renal output, on and on. So the default currently is do a large animal study in beagles, either in Toronto or in Houston, because it's currently probably the best model. But we'll see what happens with the FDA. Not sure what else to tell you about that. The way beagles handle beta amyloid as they get older, the old dog, new tricks, they show cognitive decline just the way we do, is very much similar to the way humans handle it. So it's probably a pretty good model. But again, partly it's a physiologic question, partly it's a political question, and we'll just do whatever it takes.
Yes, thank you for that response. And if you're able to achieve significant life extension in beagles through some of the treatments that you test, this, I think, will spark great consumer interest because a lot of people would be quite intrigued by the prospect of lengthening their pets' lifespans.
Right.
More
but
so.
Let me put it, provide anybody who's been involved in longevity for the past 50 years knows the traditional problem, which is the only way to prove you can double the human lifespan is to wait another 70 years. Thanks. What about now? And there we get back to biomarkers. So when we're doing studies on dogs or studies on humans with Alzheimer's, we're not directly going to be saying, can we double the lifespan? We're going to be looking back at biomarkers. So for example, in human beings, we'll be looking at protein markers in not only the serum, but the central nervous system in the CSF. We'll be looking at imaging studies. We'll be looking at cognitive studies. We'll be looking at a number of biomarkers. having said that, ultimately, we plan to intervene to reverse aging with an outcome that, as you just said, will be reversing aging. But from an FDA and a regulatory perspective, we're going after Alzheimer's and we're measuring biomarkers.
Yes, that makes sense. Thank you very much for your response. And now we have a question from Dr. Jose Cordero, who has joined us in our live chat. He wonders, what does Dr. Fossel think of Liz Parrish's experiments in injecting telomerase? Any advice on doses, intervals, timing, any other issues that could be relevant to the outcome? So Liz Parrish, of course, is well known for being the first patient to receive a combination gene therapy in an effort to reverse aspects of biological aging. The combination gene therapy was for the expression of telomerase and follistatin. She received the first dose back in 2015 in Colombia, and she has had some follow-up gene therapy since that time. What I can say is I've known her since 2015. She actually made the first announcement that this combination gene therapy trial happened on my live stream in October of 2015, entitled "How Can Life Extension Become as Popular as the War on Cancer?" And she didn't say yet that she was the patient. That was disclosed a little bit later on Reddit. But the first announcement that the trial had happened and that it was a success was made on my stream. And since that time, at least in my personal opinion, observing Liz, she does not seem to have aged. She is 55 years old. She does not look like a 55-year-old. So I think something happened, but I am not a biologist. I am not a medical doctor, so I can't opine on what happened, what worked for her. But Michael, do you have any opinions?
Liz is a delight. And I still remember going out to lunch with her in Seattle before she ever did this. And the second thing to say is that she is in that long tradition that gets enormous respect and enormous criticism, usually from regulatory bureaus, of experimenting on yourself. There have been endless scientists and physicians over the past hundreds of years, centuries, who have experimented on themselves. And so my hat's off to her for doing it. On the other hand, Liz and George, for example, George Church, from my perspective, don't truly understand how aging works in the sense that Bill and I would do it. And the other issues that we've seen has to do with how it's delivered and the efficacy, the transduction rates, the serotype choice, the actual E14, E12, how big the dose was, where it was delivered. So we'll see. I'll leave it at that. You want to join in on this?
Yeah. Liz is not a scientist. She's got a master's degree, but I am just amazed at how well she understands the science better than most scientists in the field. But you're right, she doesn't understand it as well as you and I do. But I'm just always super impressed. But there's a weird analogy that I like to think about, and that's that when the Nazis first invaded and took over France, okay, there were groups of people that joined resistance efforts and fought the Nazis. And, they were usually the young people, and their parents would say, don't do that. You'll hurt yourself. That's too risky. Just, what are you, crazy? And stuff like that. I even said this at the Boston BioLife conference just a week ago. I said that Liz Parrish is like the resistance movement. She's a war hero. She's fighting in the war against aging. I'm terribly worried about the risks she's taking and things like that. But I'm not going to be any less disappointed in her as the people were that were in the resistance. Is that they're the parents of the people in the resistance. They were war heroes too. It's like it's a big fight that's going on, and I'm glad to see people like Liz Parrish going for it. I'm not a biohacker. I would never myself do what she's doing, but I'm sure grateful that she's doing it.
Yes, absolutely. And I think, of course, different people have different risk tolerances, and I too would not subject myself to an experimental gene therapy. I've liked to say that I do not want to be in the first cohort to receive an anti-aging treatment. I would like to be in the second or third cohort, ideally the second or third generation, to receive anti-aging treatments, because by that point, the treatments will be refined, they will be scalable, they will be accessible to larger clienteles, and someone like me could then be a typical patient rather than patient zero of the first trial of a treatment. We also have joining us our friend Zach Varkaris of Austin Biotech. So welcome, Zach. We've had a fascinating conversation thus far with Dr. Michael Fossel. And I think this is a good opportunity for Art Ramon to ask a question as well about anything that we've discussed thus far. And if you have any thoughts on AI systems as well, how AI systems might be useful or not useful in your own experience, please feel free to opine on that. So go ahead, Art Ramon.
Well, yeah, I use AI extensively. I was suffering from long COVID, and yeah, I couldn't think very clearly. So I extensively use a lot of AI for decision making. But the question I had was more like blood flow. I had this device that I bought to help increase, your thinking ability, and it supposedly monitors blood flow in your frontal cortex. And I used this device, and it seemed to kind of help, uh, kind of push through times when I was tired or fatigued, and I could sort of push through with the thinking and problem solve. And that was just from using this device, which like I said monitors blood flow and has a little game on an app that you sort of control, and it helps increase the blood flow vessels or something other. Um, and that I think better vascularity in the brain. What are your thoughts on that?
I'm in favor of it. Back in 1973, I guess it was, I was in college, I actually built an EEG machine and showed that you could actually control your brain waves. And then I got intrigued with human body temperature and found that, bought a thermistor and showed that I could control not only my blood pressure, my heart rate, my brain waves, but my actual body core temperature. I could bring it up and down a degree. There are a lot of things that we can do that we're not aware of. People talk about the voluntary and the involuntary nervous system. The fact is, we all control involuntary nervous system. That's why we don't urinate all over the place. We learn to control these things. It's just a question of whether we can get the feedback to do it. And I think what you just described is something that it lets you, as you said, play a game, get some feedback on these things. So it wouldn't surprise me if you can control, for example, the blood flow. The question is the feedback accurate or not? And, are you really controlling it? I don't know, but I certainly see the potential to do that.
Yes. Thank you for that response, Michael. And we had a prior comment that I think was quite interesting. And this is from Daniel Tweed. He wrote, checking your blood pressure raises your blood pressure. Ironic. So that's in regard to patients who are concerned about their blood pressure and therefore check it regularly. But in doing so, they become stressed, and the stress raises their blood pressure. So they're not truly at rest. And I know this happened to me during the year that I had long COVID. I would get elevated blood pressure readings because I had a higher background level of stress. It wasn't really that my blood pressure was actually in a hypertensive range. It's just that I could never manage to relax fully or take a truly resting blood pressure until I overcame the long COVID issues. So I'm curious, in regard to this thread of conversation, any way for humans to kind of control these involuntary processes to the point where might it be possible to either regulate one's stress levels so well that one could always get like a true resting measure of a particular parameter, a particular biomarker, or some way to have a measurement that disregards the stress? Like, could there be a device that's developed to measure blood pressure in what would be a true resting state, irrespective of whether the body is actually active or agitated, so that you could just have continuous measurement while you're up and about doing your daily activities, exercising? I know exercise raises blood pressure in the course of exercise, so it's not a great time to measure blood pressure unless you know what is normal blood pressure for when you're exercising.
Yeah, remember that when you're talking about sort of baseline blood pressure, that is when you're most relaxed, and this is, the medical literature is full of this, both feet flat on the floor, me leaning back, my arms in a comfortable positions, upright, and then you wait 5 minutes. Okay. But the fact is that it's not the baseline blood pressure that necessarily gets you, is those peaks. The analogy might be, if I'm trying to fly over the Rocky Mountains, the question isn't what's my average altitude compared to the average altitude of the Rocky Mountains. The question is, am I going to hit that mountain on the way over? It's the peaks that end up killing people. It's the time when your blood pressure goes to 200 over 150 that you have the stroke. It's not the fact that it's 130 over 90. That's, okay, big deal. Nobody cares. It's the fact that you sometimes have that peak. So it's nice to know what your baseline blood pressure is. But in general, that's a long-term stress, but that's not what kills you in the short term. And the same, I think, can be said in regard to people who fuss too much about these things. I've got to fall asleep. I've got to fall asleep. I've got to fall asleep. You're not going to fall asleep. It's time for me to go meditate. Wrong response. So it's just, we do this to ourselves. We overstress, and that causes a problem. It's certainly true with regard to blood pressure. As I say, you get people come in and of course their blood pressure's up. You take it 10 times a day, of course it goes up. We have to have a broader view of how we take care of ourselves. Certainly not just a matter of, if I check my blood pressure, if I just relax, no. Here's an example of this. I'd find residents who would see a patient and they'd say to the patient when they're going to do a heart exam, relax, just relax. No, that doesn't make anybody relax. I found it worked very nicely when I told people a joke, because when people start laughing, they relax. Telling people to relax, don't panic, relax. Oh, I had a woman who came in, a 5-year-old, who was panicking about a suture that I was going to put through the kid. The mother is holding the baby's hand, the child's hand, squeezing it so I could see her knuckles turn white, and saying, it's okay, it won't hurt, just relax. What's the message the child got? Well, we do this to ourselves. Relax. That's not a way to take care of ourselves. A little humor helps. That standard joke, by the way, about, I don't know if you've seen this. I have to tell it just to throw a joke in here once. How many dead bodies does it take to screw in a light bulb?
How many?
I'm not sure, but I know it's not 6 because my basement's still dark. Right. Now, who is relaxed and who is stressed during that? You relax. You can't help it. It's a stupid joke, but people relax. If I told you to relax, it wouldn't have done the same thing to your blood pressure.
Indeed. So perhaps even injecting some humor or lightheartedness into one's daily routine can help with that kind of relaxation on a longer term trajectory, maybe lead people to experience fewer of those peaks of blood pressure. That's interesting. And thank you for that response. Zach Varkaris writes in our internal chat, don't sweat the small stuff. So thank you, Zach, for that. And we have a few comments on this subject. John H., More Life Please, writes that he does 5 to 7 readings and takes the average. Almost invariably, the first reading is the highest, sometimes significantly higher than subsequent readings. And he also describes the condition of artificially elevated blood pressure as white coat fever. I've, of course, heard that term as well. Some people are just made very tense by doctors, nurses, or dentists, but it's not so much even by doctors as such. It's by the act of having one's blood pressure taken, by the act of like being subjected to a test in the present moment that leads some people, I think, to stress out unless they can find a way to genuinely relax. But definitely an important conversation for those who want to avoid those acute moments of extreme blood pressure that could lead to a stroke or other adverse health condition. So now we have some questions that David would like to ask Michael about. And David, please go ahead.
So I want to thank Michael for some really fascinating words in this conversation.
David apparently can't get the lights on in his basement either.
Testing. Is this working?
Yeah, we can hear David. We can hear him.
All right. So I want to ask about the book, Michael, the Reversal 2026. And I remember coming across your previous book, or one of your previous books in 2015, the Telomerase Revolution. So I want to know what's changed, what's different. Is this simply that you're 11 years older and wiser, or has there been some significant developments that have caused you to write another book? In particular, I'm wondering about something that has happened in these 11 years, something that's got a lot of attention, which is epigenetic reprogramming, which a lot of people are sort of saying the same things that people used to say about telomerase, which is that telomerase might be the master conductor, and other people are now saying, no, it's epigenetic reprogramming. That's the master conductor. There's a lot of investment with Altos Labs and so on. It's the same thing, is it? Yeah.
Well, yes, it is. Back to the comment that Bill was making and I was making earlier. If you wanted to say what's the key to aging, I would say it's the genetic expression, that is the epigenetics. And the question that Bill and I and a lot of other people have been asking is, what's the most efficient way to optimize epigenetic expression? So again, you can use Yamanaka factors, you can attack methylation, you can go after a specific protein. Again, our question is not do those work, it's how do you make them work effectively, and what is the optimal way? Again, both clinically and financially. If to take the orchestral example, I could go to the strings only and say, I want you to play Che sapete Mozart. But it's more effective if I have the entire orchestra play Che sapete. It's not a question of just one instrument or one section. It's a question of how do I get the entire orchestra to play together in the correct rhythm, with the correct tempo, with the correct cues to each other, which the maestro will give. So it's, epigenetics, or the expression of each gene, of each instrument in the orchestra, is critical. The question is, how can I do it most effectively? What's the optimal point of intervention?
And you feel that in the 11 years since you wrote the book, there's more types of interventions that need to be considered. There are new tools and techniques that weren't possible back in 2015?
Well, that's true. Again, if I'm looking at just the example of AAV interventions and serotypes and promoters, we've changed a lot, particularly with regard to serotypes. And it's still changing dramatically from what we've done in 11 years ago. Let alone in 2001, as I say, we had an offer to take all of this translational work in 2001 with more money than you would believe. And it turned out that the people who offered all this had the money, but they got into a messy divorce. They hated each other and it dropped it. But we wouldn't have been able to do this in 2001. We would have made too many mistakes, as occurred with the early gene therapy work, just a few years before that.
Jesse Gelsinger.
That's exactly what I'm thinking.
Yes. So, Michael, I think this is a great bridge into a question that I've been curious about, since you have observed the progress of the longevity field for many decades, really since the 1970s when you were in university. how would you characterize the trajectory of that progress relative to what your expectations were, say, in the 1990s or the early 2000s, compared to what has been achieved up through our time, through 2026, what has yet to be achieved, and how optimistic are you that we who are alive today, who are here on this live stream, could personally experience longevity escape velocity?
I think probably everyone in this call, I know Bill and I, recognize that science is not just science. There are fashions and things that come in and out of vogue. And I'd like to say it's all pure science and rational and logic, but it's not quite that way. In 1996, when I first put out that book, Reversing Human Aging, I was all over the world. I was on Good Morning America. I was down to the NIH. I was everywhere. And I thought things would happen pretty quickly. And they did not. And I don't think it was partly because the science wasn't ready, but it was also because things come in and out of vogue, and telomeres are sort of in vogue and then out of vogue and then back in vogue and then out of vogue. Things come and go. And with that, funding, as well as, what does the NIH and the NIA and the NSF think about, the FDA think about these things. These things come and go, and they blow with not a big wind, but there's some breezes involved. Now, the other thing is that I am optimistic. But part of what hasn't changed is a point I made at the NIH back 30 years ago next month. And I don't know if any of you have ever been in the NIH auditorium, the old one there, but it's very steeply raked. And so people are physically looking down on you, but they're also kind of conceptually looking down on you because the topic was reversing human aging. And I knew what people thought about that, what most of the public thinks about that, which is it's impossible. So I started with a comment that I think of as radical honesty, which is I said, listen, any of you who leave this room in an hour and think you can reverse aging, you're naive. But any of you who leave this room in an hour and think you can't reverse aging, you are just as naive. If you are sensible, you'll leave here in an hour saying, show me the data. Now, let me tell you what the data there is. There isn't much, but here's what there is. I'll talk about it. I think that's the right attitude, and that's still where we are. Bill and I don't know that we can reverse aging. I don't know I can cure Alzheimer's disease. And the question is, what happens when we try it? So that hasn't changed. That's still the truth. That's just radical honesty. It's not a question of theory and debate. It's a question of clinical trials and data, not debate. But I think we now have the tools to do this. And we now have, many of us, the funding to take these to actual clinical trials and see what works and what doesn't. And the answer is the same one I said at the NIH 30 years ago, which is show me the data. And so, yes, I think we'll be able to pull this off within the next few years. Yes.
Excellent. Excellent. So perhaps within the next few years, it will be possible to answer that question, can you do it or can't you do it, with much greater confidence, depending on what these trials show.
Yeah, one of the key questions that Bill and I have seen, me for 50 years, but I know Bill for almost that long at least, is that question of is aging a disease? And to me, this is angels on the head of a pin. I don't, disease, not disease. What I notice is that most biologists who will tell you it's not a disease are dyeing their hair, getting Botox treatments, and having their knees replaced. So it's not a disease, but you act like it's a disease. Make up your mind. I don't care. And the same has been true with the FDA. This issue of whether the FDA will accept aging as a disease or not, to me, doesn't matter. Ultimately, I need to measure something. It's easier for me to go after Alzheimer's and measure imaging, measure lab studies, measure cognitive function, and some other interesting things too, like evoke potentials, God knows what. But to me, the question is, can I actually affect things that matter to people? And if they want to call it a disease, that's fine. If they don't want to call it a disease, that doesn't matter to me. I'm still going to go ahead and do it. But yes, I think we can reverse Alzheimer's, cardiovascular disease, and the only effective way to do that is by reversing aging. And whether the FDA says that's aging or not, it's not my concern. You let me know, and I'll do it your way.
Mm-hmm. Yes, thank you. And Bill is applauding your response, Michael. Now, on the subject, I know this may be a kind of hair-splitting question in your view, but if aging is a disease, and I think a lot of us transhumanists are inclined to view it that way. Daniel Tweed wonders, should we view it as a singular disease or a collection of age-related diseases? Is there just one fundamental aging disease if it's a disease, or are there multiple aging diseases? Could each type of aging-related damage be considered a disease unto itself, like mitochondrial mutations or intracellular waste? So in Aubrey's framework, the 7 strands of the SENS approach, the 7 types of damage, could they each be their own disease?
I'm going to reframe the question, but I'm going to answer it. I think I'm going to answer it by saying it's a single disease, but I'm going to reframe the question. One of the questions that comes up is, say I've got a patient with Alzheimer's and a patient with Parkinson's. Classically, the patient with Alzheimer's has cognitive deficits, but no motor deficits. And the patient with Parkinson's has motor problems and no cognitive problems. That's not true. There overlap. Many patients have both. Some patients have both at the same time, and it's hard to tell what's what. But the same is true of frontotemporal dementia and all the other alphabet soup of dementias. You find, and most of them have a vascular component as well, certainly on autopsy, if not diagnosed before that. So you get this question of, well, is Alzheimer's a single disease compared to Parkinson's, for example? And the answer is, how do you want to define it? Let me give you a crazy example of this. One of the world's most famous physicians was Ibn Sina. We in the West would call him Avicenna. And about 1,000 years ago, he put out a medical textbook that was still in use and in print 750 years later. Remarkable physician. And he knew everything there was about plague. Very good at, didn't know what caused it, but boy, was he good at diagnosing it. Now, when it comes to typical plague, you've got bubonic plague, pneumonic plague, and septicemic plague. And they present a little differently. They have different fatality rates. We treat them slightly differently in how invasive we are, but they're the same disease. So if you say to me, is septicemic plague the same as bubonic plague? The answer is, what are you really asking? You mean, how am I going to treat it? Or do you mean, what's it caused by? I think the same thing is true of age-related diseases. Ultimately, yes, I can say you've got Alzheimer's and you've got cardiovascular disease, but you also have some vascular disease in the central nervous system too. So are you dying of a vascular disease or a neuronal disease or a microglial disease? But to my mind, they all share a common stage. And what comes on stage, what goes off the stage may be a slightly different play, but the stage is still the exact same stage. And that stage is cell senescence or cell aging, changes in telomeres and changes in epigenetic expression and changes in the turnover rates, for example, with regard to mitochondria. If you're looking at mitochondria, what you find is the rate of turnover of aerobic enzymes, for example, actually a whole host of them, whole collection of them, they slow down with age. And they slow down with age relative to telomere shortening back in the nucleus, because most of the aerobic enzymes we have aren't produced locally or even coded for genetically within the mitochondria. They're imported in a sense. Not all of them. The ones that have a fast turnaround time, for example, if I hold my breath, those respond quickly in the mitochondria. They don't depend on back in the nucleus. But most of them are imported. And the turnover rate slows down with age. So what you find is that the ATP to ROS ratio goes down with age, because instead of replacing the aerobic enzymes, they're sitting around a little longer, not getting replaced as fast. And so the degradation that occurs from oxidative damage manages to accumulate. So the efficiency goes down, ROS goes up, ATP goes down. And you also find the same is true of the lipid membranes of the mitochondria. They're not turned over as fast. So because of that, they tend over time to get leakier. So you've got more free radicals, they're escaping more. And then if you look at things like catalase and superoxide dismutase, they're turned over more slowly. So you're not scavenging the reactive oxygen species, so they cause more damage, for example, to proteins elsewhere in the cell that also aren't being turned over as rapidly. So, all of those things, including the DNA repairs, they all are occurring in almost any senescent cell you're looking at. Again, my toes are different than my renal podocytes, are different from my pneumocytes, are different from my microglia, but the stage is the same. In all cases, you're looking at changes that have to do with shifts in epigenetic expression that slow down recycling rates in cells, and they all are orchestrated in some degree by telomere changes. So is aging a single disease? If you mean at a cellular level, yes. If you mean, do they present differently clinically? Well, yes, they're different diseases in that sense, in the sense that, do I have osteoarthritis or do I have renal failure? Well, that's very different. Thank you. I treat them differently. But ultimately, the stage is the same.
Yes, thank you, Michael, for that response. And we have two questions that are relevant here. First, Zach Varkaris asks, in your opinion, why do we age? And he says, forgive me if you've answered this earlier as he joined late, but Kevin Bransaforti also wonders, could human aging be a preset event built into humans during our evolutionary development? So I know there are various schools of thought on this, and one school of thought is, no, this is essentially a case of evolutionary neglect rather than a preset built-in process, in the sense that once people have reproduced and have had time to rear their offspring, natural selection stops protecting them anymore, but it doesn't actively destroy them. But others do think there's some sort of aging program at the genetic level that gets activated, and they think that if that's the case, then finding some way to edit that program out of the genome could potentially help to undo biological aging or prevent it. So what do you think along these questions? Why do we age? And is there any sort of built-in aging due to evolution, not just the result of evolutionary neglect, but something that is programmed into the human genome?
Boy, does this open a can of worms. One of the discussions that comes up again and again is aging programmed? And yes, it is. No, it's not. And to me, this is like saying, is aging a disease? It really depends on what you mean by programmed. What are your preconceptions? What are your biases? What are your assumptions? Stop calling it that. Let's get down to the nitty gritty. That is, let's look at the actual biology of it. Never mind what you want to call it. It's amazing to me how many people agree with something, but they call it something differently, so they argue. It's the old S.I. Hayakawa, the linguist, who said cow one's not cow two. If you take a common term, you find that people aren't talking about the same thing. So back to a little reality here. First of all, aging clearly is not a matter of just information loss. A couple of professors I know aside, because otherwise you'd have to explain why it is the germ cell lineage didn't disappear 4 billion years ago. Clearly it didn't. And this is the question that came up in telomere biology too. When we first realized, thanks to Alexei Alovnikov, that telomeres shorten, they do shorten over time, you got a couple of options. Either they kept shortening, in which case life disappeared 4 billion years ago, or somehow they get relengthened. Yes, telomerase. And Barbara McClintock's telomeres, and then on to the Nobel Prize 2009 with telomerase. Clearly things happen. So you can't just wave your hands and say, again, it's entropy, it's information loss, without saying how come it doesn't get lost sometimes. And it brings it back to this whole issue of programming. I skip the programming part and just try to get down to the nitty gritty about what we know. For my part, one, I don't know why we age. And I would not say it's because of telomeres or because of epigenetics. It's a more complex system than just any particular name you want to put on it. It's like saying, why does the airplane fly? Well, it's the fin belt. Well, it's the engine. Well, it's the no, it's not any of those things. It's all the whole thing together. It's a system. So when we're talking about aging and programming, I think in a teleologic sense, and again, I'm out on a limb here because I'm running experiments with evolution. Back to my description of the two sets of deer on two different islands, one of which has a 1-year lifespan, the other has a 500-year lifespan. The 1-year lifespan will respond faster to environmental changes, whether it's predators, prey, biology, temperature, pH, and anything, global warming. The shorter the lifespan, the faster the turnaround time. In that sense, aging is very, very useful from a species perspective, not from your perspective, but from a species perspective. And likewise, as I said earlier, the same is true of mutations. You do not want a biological system that never mutates, or the environment will change and it'll die. You do not want one that is mutated all over the place every second, because it'll also die. Not only do you want just the right amount of mutation, but if you're smart in a godlike teleologic sense, you design a system that is responsive to the amount of change in that environment. And I think that system is exactly what we're seeing in terms of genes epigenetic expression, telomere shortening, and everything that goes with it.
Yes, thank you for that reply, Michael. And it does seem to be a complex process, of course, and there may not be a singular answer to why we age. I am curious in regard to, let's say, the biological or even the mechanical explanation for why telomere shortening leads to a lot of these aging associated processes and dysfunctions. So telomeres are the end caps of the chromosomes. They're non-coding, so they don't actually express any sorts of traits. Is it just that, say, the shape of the DNA molecule becomes deficient in some way because it doesn't have those end caps? It can't fold in some manner that's useful in interacting with components outside of the molecule itself? Or what is happening there that just the shortening of the end caps can lead to this wide array of dysfunctions, just to help our viewers conceptualize it? I know Bill has some very useful illustrations that he utilizes in his presentations, but how do you view it, Michael?
Yeah, I was going to say, I'm going to start answering this. I'm going to turf it all onto Bill, let him take it. The first thing to say is that there's this common misconception, the aglet analogy, which is that, you know, after a while you lose your telomeres and then the DNA frays, the chromosome comes apart, which is nonsense. Your chromosome, the only way a chromosome totally comes apart is when you're dead and it's rotting. But having said that, the telomeres do shorten, and they have dramatic effects not only locally, and there's a whole enormous complexity of the telomere sheath that we're still not fully understanding. But it's enormously complex locally in the telomere, around the telomere, or the shape of the telomere, the L loop. It's very complicated. But beyond that, as the telomere shortens, not only does it have effects in the subtelomeric region, but those have effects broadly throughout not only the rest of that chromosome, but other chromosomes as well. So as the telomere shortens, it's having broad effects on gene expression throughout the entire cell, and probably, for that matter, some of the mitochondrial DNA. Bill, off to you.
Mute.
Oh, you're muted.
Okay, so I don't have an unmute button here on my computer. I don't know why.
Oh, that's strange, but I did just unmute you, Bill.
Yeah, you muted me a long time ago. I've tried talking several times. I mean, it's okay. So I think of the telomere as I said before, like an enhancer sequence. And the reason why, like, some animals will age at different rates than others is probably due not to the length of the telomere, but the distance that the relevant genes are from the telomeres. So the telomere is most likely folding over just like an enhancer does in a chromosome and interacting with the promoters, which I like to call dimmer switches, but they're more complicated than just dimmer switches. But they interact with the promoters that turn them on, turn them off to different levels and things like that. And the shorter the telomere gets, the less it's able to reach. And it doesn't have to reach all the way to the middle of the chromosome to be able to do this. All it has to do is to reach to the transcription factors that then go and regulate the other genes that are long ways away. And if you do an analysis of genes on the chromosomes, you'll find that like 90% of zinc finger transcription factors are right adjacent to the telomeres, the genes form. And so it kind of is suggesting that they are playing a very key role. Of course, we don't know. Fortunately, a paper did come out just last year where somebody's actually now got data showing that telomeres do act like enhancers. And I showed the reference at my last presentation. But Woody Wright and Jerry Shade published a lot on telomere position effects without exactly knowing why. But the enhancer role of telomeres plays that. Enhancers, the telomeres have the same histones that bind to the regions that enhancers do. They have several transcription factors like molecules like TRF2 that actually do bind and do regulate genes. TRF2 has been shown to regulate several different genes, and TRF2 is a protein that binds specifically, most often, to telomere sequences. And so it's like, it's not a question, are they enhancer sequences? They are, because the structure and function says they are. The question really becomes, is there any data that say they don't act like enhancer sequence? And so far, there's none. So I do believe that, but on the subject, going back a second, we were talking about I think it was Daniel asked, is aging accumulation of diseases or just one disease? And I agree with Mike. In fact, I haven't disagreed with anything Mike has said during this whole thing, but it's like,
oh, that worries me no end, Bill.
Yeah, well, we'll find something.
We'll find something.
Yeah, when I was in college, I was studying a lot of statistical theory, even though it wasn't part of my biology degree. I took it in experimental psychology, just because I was trying to answer that very question back then. And the fact is that, yeah, there's a lot of diseases that affect our health and therefore affect our lifespan. But do they affect aging? Trying to separate aging from health is a really big issue. But we can tell, and this is what I learned back when I was doing the statistical theory classes, is that the distribution of the age that we are when we actually die is too narrow to be explained by an accumulation of multiple diseases. If it was multiple diseases, we would see a much broader curve. Now, there are people that die young and people that die old, but the actual curve, the whole curve of everybody, is just way too narrow. In addition to all the diseases that cause us to die prematurely, there is one disease that is actually controlling us like a clock. And then it probably is playing a major role in controlling all the other diseases and controlling the epigenetics. And so far, I have not seen any better explanation than telomeres.
Hey, let me add two pieces to this. Some misunderstandings people get. And it's something that came up when Bill was talking about this. A lot of people say aging, it can't be explained by telomeres because, for example, some mice have telomeres 10 times longer than mine, and I have a lifespan that's 40 times longer than theirs. And it turns out there's a really good correlation between telomere shortening, the rate at which you shorten, and lifespan, species lifespan. But if you try to just correlate length of telomeres to species lifespan, the correlation goes all over the place. It's pretty poor, down around 0.2 or something. But if you're looking at the rate at which they lose telomeres, then it correlates very nicely with species lifespan, which brings us back to Bill's point, which is it's not just the telomere length, it's the effect it has as a dimmer switch or as an enhancer, as a promoter, and the way it affects gene expression. It's not the length compared to different species. Within a species, within an individual, yes. The other point that always is amazing to me in the literature, and I've, as you know, I started off the rejuvenation research or, anti-aging medicine journal. But I see this in a lot of journals I've edited. People will send in articles and say, I measured telomere length and it doesn't correlate very well with heart disease or with osteoarthritis or something else. And because they're measuring the wrong thing. What they're doing is they're measuring peripheral leukocyte telomeres, which have very little to do with my knees, very little to do with my coronary arteries, very little to do with my Alzheimer's risk. Not nothing, but not very much, because there's a correlation. For example, I have gray hair. That should correlate with osteoarthritis. I don't have osteoarthritis. If you try to diagnose osteoarthritis on the basis of the color of the hair, that's a very poor diagnostic criteria. That doesn't mean there's not a correlation, but it's not very big. But the same is true when you're looking at peripheral leukocytes. It's the wrong measure. Even if you're trying to measure immune function, it turns out it's not a very good measure because peripheral leukocytes churn. I could go into the technicalities of it and I've written a couple papers on it, but the bottom line is most of the literature is not very thoughtful. People will say mice have long telomeres. They'll say the telomere length that we measured in the lab doesn't correlate. It's good data. It's bad interpretation.
Yes, thank you for
that
response. Yes, go ahead.
I have a meeting that starts in 5 minutes. I have to go, so I wish
I could stay.
Good to
see you again.
All right.
Yes, good to see you as well, Bill. Any closing remarks from you, Bill, before we move on to some of the few remaining questions that are left?
Well, I'm beginning to understand why when I did that episode in 2021, why it went 3 hours. okay, listening to Mike talking right now, it's like, I don't think Mike has scratched the surface of all the things he can talk about in terms of aging and things like that. But it's something, let me just say that something I've been talking about recently at many conferences is and Mike brought this up a little bit about funding is the big issue. There's actually more, the gauges of success is really the big problem with funding. And it's like academic labs and a lot of commercial labs, they gauge their success on the number of publications they get. And it's a lot easier to get publications by publishing phenomena you see about telomere length than as actually trying to actually lengthen telomeres. I mean, if your only goal is to find a way to lengthen telomeres, your only publication is going to be when you actually succeed. And so you don't get a lot of publications, therefore the gauge of success is low, your ability to get grants is decreased, and et cetera. So, and then also the return on investment for investors is so low that when they have to wait all this time for you to actually come up with a cure for aging, or let's say a way of lengthening telomeres, it's going to take a long time for them to get return on investments. And investors, their gauge of success is how quick they can get return on investments. So we have a problem. We have to solve the problem of how to actually put more money into curing diseases than actually just characterizing diseases. And that's one of the big problems right now, because it's the return on investment, the number of publications that you can get is just not sufficient for when you're trying to actually cure something. And that's, I don't know, I spent in my videos, I spent a lot more time mentioning that. I probably was a lot more clear in my videos, but I need to run to this other meeting now, so I'm going to leave it at that. But we have to figure out the funding problem. All right.
Indeed, we have to figure out the funding problem. Thank you, Bill, for that. And once again, if you would like to hear more of Bill's thoughts, not just on the funding problem, but his understanding of telomere biology, some of his lifestyle approaches that have helped him over the years, watch our virtual Enlightenment Salon of October 10th, 2021. It is one of our most viewed salons, and it is indeed a 3+ hour discussion, well worth your time. So now, Michael, we do have some additional questions for you. So let us pose this one from Zach Varkaris. He wonders, based on what you've said, is aging the optimization of adaptation? For instance, within the example that you provided of the very long-lived deer and the very short-lived deer, how they adapt to their respective environments. And could you, for instance, conceive of a situation where a species evolves in a very benign environment, a non-stressful environment, slow changing, where the reproduction rate of that species is also very low? So the evolutionary, let's say, impetus would be to keep the individuals of that species alive for as long as possible, rather than have high turnover and a high mutation rate.
Yes.
Very good.
Okay.
Very good. Now, Zach, I wonder if you have any follow-up questions, thoughts, comments on this or anything else you've heard. No? All right. So I think, can then move on to another question from Miriam Abbas. She wonders, how could we be able to fully regenerate our limbs the way some lizards can, for instance? And she knows there have been some studies, but they haven't gotten too far. And it is indeed remarkable. There are various animals that can regenerate full limbs, or some animals, some worms, you can cut them in two, and the part that had a tail grows a head, the part that had a head grows a tail, and they don't die from that kind of trauma. So could any changes be engineered in humans to give us that functionality?
Yes. Now, it almost certainly is not simply telomere length. But I'll give you an example. We know that even in human neonates, if I take a human neonate, if I take a 5-year-old and they lose a finger, they lose a finger. If I take a human neonate, there are cases where we've lost a finger in a neonate and they regrow. So it's not that human beings can't regenerate, but there seem to be age-related restrictions on that. And for that matter, I've never seen a case where the human neonate has lost an arm. But fingers, yes, certainly parts of fingers. It happens. Rarely, but it happens. All I can say is there's a lot I don't know about that. There's a lot we don't know about that. And it almost certainly is not purely a telomere phenomenon. Hey, let me make one final comment about something that came up about funding on this. And there was a talk I gave at Harvard Business School about this too. We tend to have, whether we're looking at the NIH or anything, anytime you've got a big institution, their assumptions tend to be that what we should do is what I did when I started this career 40 years ago. And that lets out a lot of innovation. Now, most innovation and new ideas are wrong, but some of them are not. But there's this inertia, this organizational inertia in anything you're looking at. And it's certainly true of the NIH. So it gets in the way of new ideas. But the same is true of investors. Now, our return rate for people interested over the past 10 years in investing in us is less than 0.2%. Okay. And I'm dealing with 500 or 600 different things we've looked at. Not many people are interested in doing this for any number of reasons, partly because you can't reverse aging. Well, if you know that, if you know you can't have airplanes, if you know you can't cure polio, then it's going to be true. But we've found at least 2 parties that have come to us indirectly, sort of from the back door, who have interesting characteristics. First is they want to remain anonymous. Second is they don't want anybody to know how much they're going to invest in us. And third, I think most importantly, from my perspective, they'd like us to end up making this available widely and as cheap as aspirin. I can't make this as cheap as aspirin, but we can make it a lot cheaper than it costs to take care of an Alzheimer's patient. And it brings me to that final point I made in the book, and I'll make again now, which is that classic question Margaret Mead got asked, maybe apocryphally, by her students. And they said to her, what defines the very first instance in anthropology, very first instance of human civilization? And they expected her to say pottery, drawing of gazelles on the cave wall. She said, a healed femur. They said, why? It's because that implies that civilization was advanced enough that people were compassionate. They cared for somebody for months. They fed them, they cleaned them, they protected them. That is civilization. I think the same thing is true here. When we're dealing with doing something about age-related disease and aging, the mark of a true civilization is to be compassionate and do something about people around you.
That's remarkable that there are investors whom you describe who are willing to accept the 0.2% return and are genuinely interested in achieving the reversal of aging for as many people as possible, but they're hesitant to disclose their identities. Perhaps these are people who are either well known in other respects, but they don't want to be known for this, or they just want to stay out of the spotlight. But it's remarkable too that in our day and age, people with that kind of immensely benign and benevolent set of motivations would want to keep those motivations out of the spotlight. It does suggest culturally we have a way to go because we need a culture where that kind of motive, if it's stated openly and clearly, as we're doing here, will receive universal acceptance. The way that, for instance, stating we need to cure cancer receives pretty much universal acceptance today. That wasn't the case even, say, in the 1950s. But I am hopeful based on the remarks that you provided just now. So just a few more questions before we conclude our salon. We have one question from David Wood about the book Reversal. Who is the intended audience, and are the politicians or others who control the funding allocations hearing your message?
The intended, let me start by saying, I don't know much about quantum physics. I don't know much about a lot of things, but it doesn't mean I'm not interested. I got a book here at my desk about the lost Enlightenment, about the Maghreb, the Arab Empire, 1,000 years ago. Okay, what do I know about that? Not much, not my area, but it fascinates me. And likewise, quantum physics, and likewise, a lot of other things. I think that the audience I'm looking at is like that with regard to aging and biology. They're not biologists, they're not researchers, they're not physicians, but they'd sort of like to know if we can do anything about this, because all of us age. So that's my audience. I sometimes say it's the people who sort of went to college, although one of the brightest people I ever met never finished high school. The people who sort of went to college, they're curious about things. This is not their area, but they'd like to know. That's the market. I have to confess that I've ended up sending off probably 24 copies of this to the head of the FDA, the head of the NSF, head of the NIH, a number of people on the longevity caucus in U.S. Congress, a lot of senators. So I've sent copies of this off. But the reason that I want to do this is not because I expect them to fund it. It's because I want to build up credibility over time. I know, as I said, in a couple of years, I anticipate being able to come up with some data showing that we can actually reverse Alzheimer's. Nobody in their right mind should believe that. So I have a private understanding with the number of people involved, which is when I announced some of this in one of the AAIC meetings in a couple of years, somebody, probably a journalist, is going to set up in the back of the room and go in and say, Dr. Fossel, are you saying you can cure Alzheimer's? And I'm going to say, no, I'm not saying that. I'm simply saying this is my data, but thank you for asking. Next question, and sit down. I would like the data to speak for itself, not the debate, not the prior failures, not the politicians, not the NSF, not the NIA, not the NIH. I'd like the data to speak for itself, and I think it will.
Let the data speak for itself. I think that's a great outlook. David, do you want to add anything?
So are you reaching out to people like Evolution or ARPA-H, the others who've got potentially large budgets who might do discretionary funding for the kinds of projects you have in mind? Maybe they will help your research into Alzheimer's and beagles go faster. Yes, I have. But anybody who's got money has gatekeepers. Trying to get in touch with Bill Gates is not an easy thing, for example. Trying to get in touch with me can be hard enough. And the same is true of Evolution. Maybe most of the audience knows what that is, but never mind. It's a big funding source in the Middle East. Leave it at that. But they start with the same preconceptions, which is that aging just happens, things fall apart, it's entropy, what do you expect? They're dealing with shortgevity, not longevity. And I am. Having said that, yes, that's part of the reason I wrote the book. And I've sent copies off to a number of people who are involved in Evolution. And hopefully they'll read it and go, wait a minute, maybe we should be thinking a little more broadly, a little more deeply about what we can do. So that's part of the purpose of the book. Thanks.
Yes. Thank you, Michael. And Kevin Branciforti has an interesting question in terms of wanting to improve investments. He wonders perhaps there could be something with a higher potential return for the investors, such as the creation of bioidentical organs. So, of course, we have a shortage of transplantable organs right now. And even in terms of saving lives from acute accidents or the kinds of illnesses which kill one for lack of an organ, for lack of, say, kidneys or lungs, if it were possible to create bioidentical organs that don't face immune system rejection, that don't face the transportation issues that donated organs do today, a lot of lives could be saved, but perhaps that could be a bridge toward at least reversing parts of aging. I know there are various researchers in the field who favor a kind of replacement approach instead of reversing aging at the cellular level. Just take out an aged organ, replace it with a young organ that's biologically identical. What do you think of that as an avenue of research, as a possible way to achieve some amount of age reversal?
One, I can't do everything. I can do things I can do. Two, when it comes to replacement, if I've got somebody with advanced knee osteoarthritis, I could replace it with an artificial knee, which is what we generally do now. That's the default treatment. I could try to use stem cells. I could try to use senolytics to remove those cells, although that probably accelerates the process in the long run. Or I could do what I anticipate doing, which is to show that we can reset the chondrocytes there and grow entirely young knees without having to put something else in. Just take what you've got and reset the clock. So different approach. Now, a comment about the long-term profit on this thing. I still remember Mother Teresa never was in the red. That woman was a businesswoman through and through, but she wasn't doing that to make money. But she made sure that she made enough money that she wasn't going in the red. Smart. That's where our investors, we have 2 global groups who committed fully to investing us. Same routine. They don't expect us to go in the red, but they're not in this for us to become a unicorn, another $1 trillion company. We may do that, but that's not why they're doing it. They're doing it for other purposes. Again, they do not expect us to end up in the red, but they're not doing it because they expect a return in Q3. Nope, that's not it. They expect a return in 2 years, in 5 years, in 10 years, and more. And as I say, in both cases, they've made it clear that they would like us to assure that we can not only do this in Greenwich and in the Bay Area, but that we can do this for people in Chad, and in Somalia, Eritrea, in Sri Lanka, in Laos, people who, if I turned to you and said, listen, 3 years ago, a napkin calculation was just about $30,000 we could do this per head, okay? How many of in the audience have $30,000? How many could get it if I told you could cure your Alzheimer's? But how true is that in most of the world? Not so much, okay? No, we'd like to make sure that, it may not be as cheap as aspirin, but we're committed to taking this not just for profit, but to doing something that I think will make it a transhuman world.
Absolutely. And I think that's an excellent outlook for those investors to have. It's a more prudent, long-term oriented outlook in that they do not want a loss, but they don't insist on profits right away. They recognize the long-term nature of this endeavor and the transformational potential if this is done right. Now, we have one more question from our Director of Longevity Outreach, Ben Ballweg, and then we will go into any closing comments. So Ben wonders if you could tell us the story of starting the Journal of Anti-Aging Medicine, also known as Rejuvenation Research, and why it continues to be an important publication today.
That's right. We saw that earlier. There were a couple of people that Marianne Liebert went looking for, and some people turned her down. Some people suggested me. They came to me and I agreed to do it. It's an important publication in the sense that, we changed the name from Journal of Anti-Aging Medicine to Rejuvenation Research because it was a more, credible name. Throughout this field for the past 50 years and longer, you tend to find kind of people who believe anything can happen with just the right vitamin and people who believe nothing can happen. And they disagree with each other. And there are very few people in the center who think you can do something about this, but you have to do it thoughtfully to figure out how it works. Well, that was really the purpose of, in this case, Rejuvenation Research, is to try to find the people who think you can do something about it, not because it's magic, not because it can't be done, but because it can be done if you think about how to do it right. Having said that, there's a problem with any journal like this, and it's true of Rejuvenation Research too, which is the articles, and Bill alluded to this in passing too, it's easy to publish papers. The question is, are they important papers? And I find in editing, and I've had recommended, there's a friend in China who I recommended be head of a journal, and he said it's going to be a lot of work. I said, it's not the work, it's the psychological stress of having to accept papers that aren't important, because otherwise the journal can't publish. This is a problem in all journals. Maybe not for the classic science and nature, although people get so caught up in publishing papers just because it's in science and nature. But it's a problem finding an article that actually means something. That's not easy. And that's the kind of a struggle that Rejuvenation Research has too. It's easy to find the papers in there, but you want things that actually make a difference. This is not a criticism of Rejuvenation Research. It's just a fact of life about publications in this world, and that, for that matter, books too, my book or otherwise, and for that matter, podcasts and so on. It's not easy to find things that actually make a difference. Same thing's true of investors. It's easy to invest. It's hard to find something that makes a difference. That's the way it is.
Yes. Well, it seems to me sometimes one cannot quite foresee in advance what exactly will make a difference, because there could be a particular research finding. It seems minor or incremental on the surface, but somebody could utilize it in some creative, hitherto unforeseen way, and that could contribute to a major breakthrough. So I do commend the Rejuvenation Research Journal for its openness to publishing a variety of papers. It has certainly made substantial contributions to the field. So, Michael, thank you very much for joining us today for an extensive conversation. You've addressed questions on a variety of topics related to aging and how it could be reversed in a way that will create a new paradigm for our lives, for our civilization. Again, we are talking about longevity, not shortgevity. Are there any final thoughts that you would like to share with us? Anything that perhaps you would like to cover in addition to what has been said? Anything you would like to reinforce for our audience? And any more comments on what you plan to be doing next?
I'd like to say I've got something wise to say, but I don't. Maybe that's wisdom itself. Best I can say is that I think that if all goes well, and things never go well, that we'll be in human trials within the next 2 years. We'll be able to demonstrate this within the next 5 years, and not just in Alzheimer's, but we'll be spreading out throughout multiple organs. So yes, I think that within 10 years, we'll be able to do something remarkable. Again, that depends on a lot of things I can't control.
Yes, indeed. But it is definitely a hopeful plan, and one that we should all be interested in materializing. We have a lot of compliments for you from our audience. Mike Lazine writes, "Thanks, Dr. Fossel, for joining us. Come back many more times in the future." Jason Geringer writes, "This was a good one. Well done." And Daniel Tweed says, "Great work to Michael Fossel." So thank you, Michael, for offering us your time, your insights today. This will definitely help many of us, hopefully as many of us as possible, to live long and prosper.
And prosper.
Indeed.